Okay, so we're going to get started. It's a beautiful fall day outside, and our first speaker is from the Division of Cancer Epidemiology and Genetics. It's Neil Caporaso, and the title, Epidemiology. Neil, take it away.
Thank you. It's great to be here, and welcome to all those watching electronically. The title of this is Epidemiology, or Why the Governor of New Jersey Should Not Make Your Public Policy on Ebola. Okay, so just by way of introduction, I live at NIH, specifically at the National Cancer Institute, and I want to point out that a number of the other institutes also are interested in epidemiology, that is in population studies. And of course, we are intramural. 85% of the Cancer Institute's funding is extramural, but we are uniquely intramural, and our division is focused on cancer epidemiology and genetics. That is the cause of cancer. And our branch is the genetic epidemiology branch. We focus on genetic components to the etiology of cancer, but we have other branches that focus on nutrition, hormones, infectious disease, occupation, statistical methods, and radiation. So I'm going to give you the population perspective on cancer, and I'll give you a few foundations. I'll highlight some tools that epidemiologists use. I'll tell you a few accomplishments of population scientists. I'll tell you some of our challenges, and I'll point out some future directions for this area of science. And I hope to run through this lecture a little bit faster than usual, and I will certainly leave time for questions at the end. So some introductory concepts. Epidemiology is the science of the causes of health and disease in human populations. So you can see the Greek derivation here, but a key point is that epidemiology is an observational science. Unlike experimental sciences, you don't get to pick in epidemiology who are your exposed people or who are your unexposed, unlike an experiment in an animal laboratory. People who are out there make choices about what they're exposed to. They choose to smoke or to consume alcohol. And so there's always this factor which introduces some bias into epidemiologic study designs, and we'll talk about that a little bit. Epidemiologists are ethically prohibited from doing experiments on people, so we observe large populations, divide them into groups, and then assess their outcomes and use statistical methods to relate those outcomes to factors of interest. And this is historically important because early in the study of lung cancer, and I'll go back to lung cancer often as a key theme, the idea that tobacco was the cause for lung cancer was vigorously fought by the tobacco companies. So what are the goals of epidemiology? We want to understand the causes of disease. Once we understand the cause, we want to quantify those risks. We want to identify risk groups. We want to understand something about the mechanisms. It's not enough to say tobacco causes cancer. What is it in tobacco that causes cancer? We use epidemiologic research to gauge public health efforts and costs and budgets, and we'd like to identify syndromes. Epidemiologists typically emphasize prevention. It is much cheaper and more effective to prevent a disease than to treat a disease. So this creates a little bit of a paradox in that politicians, who are the ones that provide the funds to deal with public health, find that curing disease is a much more emotional, appealing, and immediate form of gratification. And so if you say, we have a new cure, you tend to get funding much faster than if you say, look, if we reduce the saturated fat in hamburgers by 0.2%, in 30 years we'll prevent 100,000 coronary infarctions. Well, that may be true, but it's just not as appealing, and so politically it's a little bit of a conundrum for epidemiologists. Nevertheless, there's no question that prevention is the way to go. Epidemiologists worry about bias. I told you that as a side effect of not being able to do an experimental design where I place the rats that I've exposed in one cage and the rats that are unexposed in another, but I have to go into a human population and I have to assess exposure. Some people may be exposed, some people may not. I'll give them a questionnaire. The questionnaire may be slightly inaccurate, there may be misclassification. So for a lot of reasons, epidemiologists are very concerned about different kinds of bias. And an example is selection bias, which means that the people in your study may not be representative of the entire population or the group that you're most interested in. And this results in a problem because then your results may not be generalizable to the larger population. So a number of years ago, we did a large case control study in Italy, a study of lung cancer. And I mentioned before that we're in the intramural program, and the intramural program has site visitors. These are outside scientists that come and review our program. And the site visitors came and said, gosh, you're going to spend millions of dollars. You proposed to study 2,000 lung cancer cases and 2,000 lung cancer controls. Are your controls representative? And they said, well, you know, if you don't have at least a 50% participation rate, we're not going to let you do the study, which, you know, was a pretty big deal. So we did a phone survey. We called up people on the phone and said, hey, if we give you $20, would you let us ask you a questionnaire? It'll take about an hour, and give us 70 cc's of blood. That's about 4.5 tablespoons. What do you say? Well, less than 30% said they'd participate in that. No study. So then we sent them an invitation letter. We followed up by phone. We went, if they were in the hospital, we went there. We made advertisements. We increased the cash award. We got a letter from their physician. And we would study them either in the home or the hospital, wherever was more convenient. Response rate, 49%, still too low. So then we got extremely charming and personable interviewers. We had their physicians call them up. Instead of money, we gave them a gas coupon, which was a big deal in Italy. We had TV ads by who was the Johnny Carson of Italy at the time. We got a letter from the mayor. We got an invitation letter that was written on a 6th grade level, and we gave them a toll-free phone line. At that point, we achieved our response rate that was needed. So this is the kind of thing. This took us a year, and I don't even want to tell you how much it costs to do these kinds of studies, but this is what's required to get a population study that's reasonable. Controls. If you ever do a population study, you're going to hear it about your controls, and this is because many small clinical studies looking at something or other will say, let's just use the folks in our lab, or let's just take everyone that, you know, is standing around here. And those so-called convenience controls invariably introduce bias and are problematic. So if you consult with your epidemiologist, he's going to look at your controls and say, they're no good. We need new ones. And population controls are the gold standard. And this is because they're most representative of the population from which your case is derived. And so they're most likely to give you valid results. And there are also some other bonuses, like you can calculate absolute risks instead of relative risks, and you reduce selection bias. So if you decide you want to do a population study for some reason, and you go see an epidemiologist as a consultant, they're going to ask you questions about your study design. That's really always the first question. As I said, they'll focus on your controls. They want you to collect covariate data. So this is super, super important. Covariate data just means factors that can influence either your exposure or your disease. And so these, if you don't take them into account in your design or your analysis, and there are two opportunities to do that, your results will be distorted. Power calculations are super important. Your study must have the right size to be able to make statistical inference about what you want to study. And there's simple formulas to do this. And typically, if your study includes biomarkers, the epidemiologist will ask you, how did you validate your biomarker? This is critical. So one other point I can't help but bring up is most common question I've gotten over many years, and that is, I have a grandmother who smokes, and she drinks, and she never exercises, and she eats buttered toast with sausage, and and she has buried 3 of her doctors. So how do you explain that? And this is the fact that epidemiology is a probabilistic science and not a deterministic science. So anything can happen to any one individual, but we're worried about a population. And so it's a probabilistic versus a deterministic issue. And scientists, I would say, are not as likely to ask this question, but when you speak with the general public, that's here. So, okay, so what kinds of tools do epidemiologists use? And I'll just touch on a few. One of them is a cancer map. This is something that we used to do a lot more of, but it's a very handy way to look at the distribution of disease and say something about risk factors. So here's melanoma, and I think it's pretty clear that you can see that the red area indicating high levels of disease is across the southern tier of the country, and that is concordant with the fact that sunlight is a key risk factor for melanoma. Cancer maps have evolved into something else, which is GIS systems, which is geographic information systems, which are satellite-aided methods to establish databases and infer risks and learn something about exposure. Another tool that epidemiologists use is SEER, which is Surveillance, Epidemiology, and End Results. And the SEER program covers about a quarter of the U.S. population and gathers all sorts of data on incidence, survival, patient demographics, and features of tumors. So this is a tool that's commonly used. And there's a website that you can go to, and if you want information on a specific tumor type, you can get a lot of information, graphs, and publications. And I will not spend a lot of time on this, but I'll show you that if you look at SEER data, you will see a lot of interesting things. And here's an example of a bump in the cancer rates that was observed in men, but not in women. And so you wonder what exactly was going on here. And here it is. When we break it out into specific cancer types, you can see that the other cancer types are relatively smooth, but prostate shows a big bump. And what is this due to? It's due to PSA screening, which caused overdiagnosis of prostate cancer. So in a sense, this was an artifactual increase in the rates of prostate cancer. So you always have to take into account diagnostic modalities when you look at changes in cancer rate. And many epidemiologists say, well, if we're going to look at a cancer rate like an incidence, at the same time, maybe we should look at mortality to see that those two are linked, to make sure that it's valid. So here's some rates in women, which are relatively constant. Here are some rates in by race, and you can see that that prostate cancer bump in white men and African American men. And I'd like to point out that the overall rates of men and women seem to be leveling off and this is at the end of the last millennium. And why is that? And this is one of the basic and fundamental features of epidemiology. This is because of the decline in smoking. Okay, so you can see that what has happened over time is that lung cancer rates are rolling over. Okay? Many of the other cancers are relatively stable. Stomach cancer has been declining for decades. But what is driving down cancer death rates is the fact that we're getting less lung cancer because we have less smoking in the United States. Okay, so I'll just make one or two more points here, and that is that if you have incidence much higher than mortality, one of the features that could tell you is that you have a tumor that's responsive to treatment, and that is exactly what we have for a variety of pediatric cancers, especially leukemias. Okay, so one of the issues in epidemiology is that we find associations, statistical associations. And a key question is, so you found an association, but we all know correlation does not indicate causation. So how do you prove it? What types of evidence do you pull together as an epidemiologist to implicate a association as a cause? And so there are a number of them. First, if something is causal, it helps if it confers a high risk. Second, it should be consistent across different studies. Third, you should see a dose response. Fourth, there should be a correct temporal order. The cause should come before the disease, obviously. But that is an issue in terms of study design. And finally, we'd like to have a mechanism. The biology should make sense. So here is some of the data that was marshaled to establish that tobacco was the cause of lung cancer. First of all, in cohort studies, you see remarkably constant and consistent data in that the more cigarettes smoked per day, the higher relative risk of cancer, and it's extremely consistent across 3 very large cohorts. You also saw that temporal relationship. So as males began to increase their smoking in the start of the 19th century, you saw male smoking rise, and males, after a lag of 10 to 15 years, male lung cancer increased as well. The same thing with female smoking. You also saw animal studies, and these were studies by Oscar Auerbach, who was in Louisiana and did studies in smoking beagles. And he showed that the same changes of dysplasia and metaplasia that you saw in the human respiratory epithelium was duplicated in the dogs that, in the experimental dogs. And this was the kind of experimental evidence that was considered a very compelling complement to the population data. And finally, a very important piece of evidence, you saw that in the cohorts, when you established that somebody quit smoking, slowly over time, their relative risk of lung cancer steadily and consistently dropped, so that they asymptotically, this is 2 and this is 0, approached a relative risk of 1. Never actually gets to 1, but it clearly drops, indicating a nice piece of evidence that it is actually the tobacco that is causal. So what have epidemiologists accomplished? And I've just picked a few items. They've certainly identified a lot of the general and specific causes of cancer. They are advocates of public health and prevention. Again, the most cost-effective and broadly effective means of eliminating cancer. I've used the example of tobacco as a causal factor in lung cancer. And I'm going to say something about secondary tobacco smoke, because that's something we all benefit from every day. So it was the studies of epidemiologists that established that secondary tobacco smoke was a risk factor for cancer, which resulted in a lot of the public health laws which keep people from smoking in places like this room, and for which I'm sure we're all grateful for. And finally, the rise of molecular epidemiology, I'll say something about that. So I'm not going to spend so much time on the general causes of cancer or the specific causes. It's something that, certainly you can ask questions about, or we can, you can look this up. It's really, but I just do want to touch that age, environmental factors, genetic factors, and combinations encompass the general causes of cancer. And roughly in thirds, a third of cancer due to tobacco, a third due to diet, and a third due to pretty much all other factors combined. Most cancer is due to the environment, and the evidence for this comes from broad international studies where we look at international variation in cancer rates, and what we see is that the high to the low ratio is strikingly high across different populations. Now, you may say, well, Neil, that could be genetics. These different groups are widely disparate, and maybe some of them have at-risk genes and others don't. And genetics may contribute a certain amount in some cases, but another class of epidemiologic studies show that it's mostly the environment. And what kinds of studies are those? Those are migration studies. So when people migrate from a high-risk area to a low-risk area, within a generation they acquire the low-risk rate. So this is an example of a high-risk area in China, Zhenwei province. And our group has done studies there. And what is their risk factor? It turns out to be indoor air pollution from burning smoky coal and indoor cooking and ovens that are placed under the beds. So a variety of these factors, indoor and outdoor, create a very, very high rate of lung cancer in this particular region. Another example is a copper smelter in Montana. And actually, I had a cancer map, which that slide may have dropped out. But on the cancer map, there was a little red area in Montana. And that particular copper smelter spewed arsenic into the local environment, and arsenic is a lung cancer risk factor. And so the smelter was eliminated, and the hotspot over time disappeared. Okay, so back to tobacco. Tobacco is still the major cause of preventable morbidity and mortality, and although rates of smoking in the United States are slowly declining, worldwide they're increasing, and this means there's going to be more lung cancer, more cases of lung cancer over the next century. Tobacco accounts for a fifth of all U.S. deaths, millions of deaths every year, 30% of all cancers. All the cancers caused by tobacco are difficult to treat, terrific costs, and in spite of the fact that knowledge about the risk that tobacco is an extreme cause of health risks, almost a fifth of American adults continue to smoke, and teenagers continue to take up the habit. And this shows the per capita decline in the use of different forms of tobacco, but this has now somewhat leveled off. and I told you I'd say a word about environmental tobacco smoke. And the first studies were in never smoking spouses of smokers. And they established that the relative risks to these spouses was in the range of 1.2, 1.3, up to 1.6. Little by little, evidence accumulated. There were meta-analyses. And these studies, as they accumulated, indicated a number of things. First was that there was no lower level of risk. In other words, even though you were exposed to not that much, you still acquired some risk. So having a non-smoking section in an airplane with a little fan really didn't help you that much. You were still being exposed. And if you were a pilot or a stewardess on that plane, you were certainly going to encounter, terrific risk over time. So these kinds of studies began the movement to create legislation, clean air legislation, that eliminated smoking from the workplace, from schools, from hospitals, and eventually even from restaurants, bars, and many other public places. And so we really have these epidemiologic studies to thank for the fresh air that we get to breathe most of the time in most places. Okay, so I'm not going to go into many of the other risk factors, but alcohol is the number 2 carcinogen after tobacco, and it's associated with a number of cancers. Ionizing radiation is a very well-known form of a carcinogen associated with a number of cancers. There have been a lot of studies of A-bomb-exposed individuals, of subjects exposed to medical radiation, those exposed to radionuclides, and occupations that entail radiation exposure that implicate associations with these cancers. And there have been studies in the Chernobyl region most recently that established overall risk and particularly risk of thyroid cancer. Non-ionizing radiation, ultraviolet radiation, sunlight is definitely a risk factor for skin cancer. One controversy is that sunlight enhances your vitamin D production. Low vitamin D is thought to be a risk factor for a number of cancers, somewhat controversial, but generally the cancer-causing risks of the sun are thought to outweigh the protective benefit. Infectious agents clearly are associated with a number of cancers, and I won't go into it. There's been a lot of study recently in our group looking at HPV and oropharyngeal cancers. And this is an area where molecular epidemiology played a key role and essentially established that for cervical cancer, 100% of cervical cancer is due, except for some variant histologies, is due to HPV exposure. We've done studies looking at other infectious agents. For example, H. pylori shows no association with lung cancer, but it does show a strong association with gastric cancer. Although study design is critical there because if you do a case control study and look at tissue at the time of diagnosis of gastric cancer, the effect of the H. pylori actually starts to occur decades before, and it eventually reduces the cells that have the H. pylori to the degree that you can't detect them. And so you would not, if you did a case control study, just study at the time of disease, it would be difficult to discover the association of H. pylori and gastric cancer. And lately, microbiome studies are looking for associations of different organisms with different cancers. And the best established one is Fusobacterium colorectal carcinoma. But those studies are really in their infancy now. And I think over the next few years, as we begin to study the respiratory microbiome, skin microbiome, the genitourinary system microbiome, more associations will be discovered. Okay, and finally, occupational exposures are a major cause of human cancers. And here's a study of diesel exhaust. I guess one thing I would say there is that epidemiology has the issue that many of the things we study are strongly opposed by commercial groups. So the diesel, the groups that produce diesel fuel are strongly fighting studies like this that look at diesel exhaust. And so they have subpoenaed the records of our occupational studies group. They've called them in front of Congress. They've done all sorts of things to harass these studies. And so in some ways, epidemiologists, and particularly epidemiologists that have a little bit of protection working for the federal government, are your bulwarks against a horrendously polluted environment where commercial interests can do whatever they want and, you know, spew anything into the environment and, you know, degrade our health. So this is a continual battle that many people are not aware of, but our occupational studies group is continually using lawyers and, you know, fighting off the efforts of these commercial groups to stop their studies. And the second they do a study, they use freedom of information laws to get all of their data, and they hire hired guns to review the data and try and find mistakes or errors or, you know, reinterpret the data. Generally, the data has stood up very, very well, but if you didn't have government groups like ours, and I'm talking about a division, it's not my group, it would really would be a loss for all of us. And another example is formaldehyde studies that show that formaldehyde is associated with increased risk of myeloid leukemias. Okay, so what are some unique challenges that epidemiologists face? And I'll touch on 4 of them. Where is the missing E? E means exposure. So for a number of cancers, we know of no environmental risk factor. Okay, so what's going on there? What about cancers that have we know little or nothing about any cause? What about the genetic cause? So we can do calculations that show that there's a heredity which should account for a certain proportion of risk, and the genes that we've identified to date in genome-wide association studies only account for a fraction of that risk. So where are the genes hiding? And finally, new studies are extremely expensive. And so fielding a new study is not an easy enterprise. Francis Collins, the head of NIH, has advocated that we do a large new cohort to incorporate all age groups, be representative of different races, and that study would be staggeringly expensive. Our existing cohorts, like PLCO, the prostate, lung, colon, ovary study, many of these over time, and a cohort goes typically for decades, have cost as much as $1 billion. So you can't start one of these. I mean, you could buy a battleship with that much money, you know, so anyway. Okay, so I touched on these categories of cancer causation before, and basically on the gene side, there are hereditary syndromes where high penetrance alleles account for almost all of the risk, but that's rare. Most of the time, for most cancers, there are a number of low penetrance genes that confer a tiny increment of risk. On the environment side, we've talked about the different risk factors: smoking, microbes, chemicals, lifestyle factors. There's also the combination of gene plus environment. And I'd like to point out that you will hear a lot about gene environment, but there are very few studies that study this actually, because there are power limitations. one of the features is that to study genes and environment, you need to get enough subjects with the gene and the specific environment to have power to describe the risk. And these kinds of studies are rare. So I have to tell you that there are very few gene-environment interactions actually known. And I mean, I can only name one or two. I'll tell you about one in a minute. Okay, so I've told you about the gaps on the environmental side. And one example is chronic lymphocytic leukemia. This is the most common adult leukemia. And if you ask what the risk factors are, well, one of them is getting old. Okay, it's a disease of the elderly. Beyond that, we don't know too much. It's more common in men than women. There are no extrinsic environmental risk factors known. And family history is a strong risk factor. And we have been studying families with chronic lymphocytic leukemia for a few decades, as long as I've been at NIH. And we have over 50 families, high-risk kindreds, where there are two or more individuals in a family with the disease. And we have done linkage analysis and exome sequencing, and we still don't have a gene. And we've looked at pretty much every environmental risk factor we can think of, and we don't have an environmental risk factor. So it's a little bit of a challenge. I'll tell you some new directions that we're going with that one in a minute. Dietary risk factors. I just have to say, with regard to diet, although there's a lot of evidence that diet in general contributes to cancer, the precise dietary factors that are associated with specific cancers remain a little bit difficult to identify. So high caloric intake and obesity is certainly related to uterine cancer. There's controversial data that high fiber is somewhat protective for colon cancer, and meat may be a risk factor. The data for micronutrients in lung is controversial. Dietary contaminants certainly are risk factors for certain cancers. So aflatoxin is clearly a risk factor for liver cancer, and it's a much greater risk factor in the presence of hepatitis B or C. But general risk factors for cancers are not well understood, and a lot of what people think are associated, like, oh, meat, saturated fat, well, the studies are really not so clear. So we've been interested in, again, lung cancer, and one of the classic issues in lung cancer is that there was a fair degree of suggestive evidence that beta-carotene was a protective factor for lung cancer. And so an interventional trial was conducted in Finland among 30,000 smokers where they randomized the group to receive, one group to receive vitamin E and another group to receive beta-carotene. The study was called ATBC, standing for alpha-tocopherol beta-carotene. And many of you may know the result of that study, which was that the beta-carotene intervention group ended up with more lung cancer. So this showed that the role of diet in cancer is one that we just don't understand so well. So I think a lot more has to be done. We need to investigate obesity, processed versus traditional foods, meat and vegetable consumption, how weight interacts with all of those, et cetera. So here's a study we did of fresh meat and processed meat and lung cancer in Italy. And in Italy, they love processed meat like the salamis. And in this study, your intake of these meats showed a nice dose response with increased risk. But I have to tell you that there are methodologic weaknesses to this kind of study. One is that it was a case control study. A second is that smoking and other risk factors tend to be associated with meat consumption. So people that eat a lot of meat also tend to smoke a little bit more. Now, we took this into account in the analysis, but some would argue, oh, there was residual confounding that may have still accounted for some of your effect. So the question of diet and lung cancer is still controversial. Okay, I told you also that there are gaps on the genetic side, so I'm not going to spend too much time on this. And as well, I told you that all cancer is due to the environment. It's also due to genetics, and that genetic changes accompany all cancer. These are somatic changes, and some cancers also have germ-like cancers. So if you look at epidemiologic studies that focus on genetics, they either focus on inherited changes, which are in germline DNA, or somatic changes, which are the changes that you observe in the tumor itself. They tend to be family or population studies. Family studies look for high penetrance, rare genes, whereas population studies look for common genes that confer only a little bit extra risk. And these are candidate or agnostic studies. In the old days, we did candidate genes, where we said, well, I think that this specific gene is the one that's the cause, so let's look for it. But today, we do genome-wide studies, so we try to cover the entire genome and look at all of them. And this is really what has revealed the genetic basis for most cancers. Why look for an inherited basis to cancer in the first place? And the reason is that family studies clearly show that risks of virtually every specific cancer, and I've showed you an example of lung cancer here, are increased in relatives of cases compared to relatives of controls. So you can see here that having a family member diagnosed with lung cancer increases your odds ratio 1.57. That's a 57% increased risk, and that takes into account other factors such as age, gender, where you're living, your education level, a lot of detail on smoking, etc. And genetic epidemiology is the branch of epidemiology that looks at these genetic factors. And I told you already that if you want to look for rare genes that by themselves confer high risk, you need families. And I also told you that here's an example of one of our lymphoproliferative disease families with a lot of CLL. And to date, we have not found the gene. We're looking very hard. A lot of tumor suppressor genes were cloned in these families. So now I'm going to move ahead. And this just shows you an example of all the different low penetrance genes that we identify in genome-wide association studies. But I'm going to move ahead here, skip over lung cancer, and tell you a little bit about molecular epidemiology, and then I'm going to move to some future directions. So, the most basic kind of epidemiology is looking at an association, a statistical association, between an exposure like tobacco and a disease like lung cancer. But what we have done over time is evolve to a system where we don't only collect the exposure data, but we also try to collect biospecimens so that we can look at the steps that happen between the exposure and the disease. So first we measure an internal dose. So you say you smoked a cigarette, but when we measure the cotinine in your blood, how much do we really find? And if we look at DNA adducts, which are the carcinogen in tobacco, adducted to a macromolecule like hemoglobin or DNA, how much do we find there? How much actual dose do you get? And if we look at cells in your respiratory tract, how altered are these cells? And finally, can we find evidence of in situ cancer in those with exposure and disease? So by incorporating all these components, we hope to elucidate the mechanism. And this is the idea behind molecular epidemiology. So that study I told you about in Italy, we actually got tissue as well. So we would go into the operating room and wait for the resected tumor, give it to a pathologist. The pathologist would cut us a piece. We put it in liquid nitrogen. And that is a incredibly valuable resource that we have today. And we, of course, would ask a questionnaire, and going after diet, we would, of course, gather all the dietary information on what they ate, but we would also explore other features like doneness. So we wouldn't ask them how much do you cook your meat, because it's a hard thing for people to answer. We'd show the pictures. Does your meat look like this or like this? So we get a lot of information on detailed information. Molecular epidemiology has contributed a lot, and it really would take almost another entire lecture to talk about that. I'll just focus on one thing, and that is that pretty much all of the GWAS studies that have established the genetic component to virtually every tumor have been based on biospecimen collections that were incorporated into cohorts. The idea that we proposed in the 90s that you can't just give people a questionnaire. You've got to get a tube of blood or a vial of urine that we can put in the freezer, and when we develop the assays, we can study them. And, we'll get DNA from it and look at the genetics. So that is exactly what has happened, and that was a really important advance. So one other kind of epidemiology you'll hear about is integrative epidemiology. And what this says is, no, it's not only the exposure, but it's the behaviors that make you get the exposure. And in fact, tobacco smoking itself has a genetic component, and we should study that. So an example of this, and I don't think I've put in a slide to show you, is that we look at nicotine dependency, and we have a paper in JNCI that just came out a few months ago that says that, your time to first cigarette, how long you wait in the morning to smoke your first cigarette, indicates how addicted you are to nicotine. And if you're very addicted and you smoke 10 cigarettes per day, you absorb a lot more carcinogen than if you're not so addicted and you smoke 10 cigarettes per day. And that turns out by itself to be a powerful risk factor for lung cancer. At the other end of the spectrum, we look at outcome. So we know you have disease, and you're alive when you have the disease, but how long do you survive? And how do you respond to treatment? And this particular area, disease to outcome, is one where there are risk factors as well. So, for example, if you're a smoker here, your outcome is a lot worse. So that's called integrative epidemiology, and we put all sorts of questionnaire instruments to dissect the behavior part, and we incorporate treatment, survival, and prognostic and clinical information at the other end. One of the ways we do these big studies is through consortia. So there are 4.2 million subjects followed in cohorts. And this is one reason why we think that cohort that Dr. Collins is proposing might not be the answer. An alternative might be, hey, let's gather more information on these folks because we already have the cohorts already. We just have to add components to make them work better. Okay, I'm going to skip ahead, and I'm going to tell you about some future components to epidemiologic studies. Because as I told you, we don't have a lot of the exposures that contribute to cancer. And we'd like to incorporate new technologies that don't cost a lot, are non-invasive, are reasonably acceptable to people, can be validated, and can be used in large populations. And I'm going to cover 8 of these in about 2 minutes because I want to end on time. The first is sleep. Sleep is clearly a risk factor for a lot of conditions. Obesity is one, mental illness is another. And so there are now, it's rather difficult. There are some validated questionnaire instruments, but it's difficult to get a good sleep history. You can give people a cell phone and some new technologies and put it under their bed, and you can get the stages of sleep, the time that you spend in each kind of sleep, the total hours of sleep, the amount of movement during sleep. And all of these factors are interesting ones to look at in relation to disease and haven't really been examined much in relation to cancer. Physical activity. So this is a difficult area to ask, how active are you? I'd have to ask you 50 questions, and if you go to a health club, or, what about if you're vacuuming the rug, or you walk to school every day, or there's so many ways that you can have physical activity. So now there are a lot of technologies, from Fitbit to the new generation of Apple and Samsung software, which can automatically collect different kinds of fitness information. Many of you use these for Runkeeper, or there's a variety of them. And little by little, these are being refined to the point where they can calculate not only activity, but periods of inactivity. Inactivity may be as important as your activity, calories, climbing, steps, etc. Heart rate can be calculated with a number of electronic means. And heart rate variability, arrhythmias, a maximum heart rate, how you respond to diet, and how you respond to exercise are important. Social factors. So it's become clear that who you interact with can have a big impact on different risk factors. So smoking and obesity are two factors that have been shown to be related to who you're closest to in a social network in the Framingham study. So this is an area that is increasingly of interest. Location. So you saw the GIS that I showed you earlier, but your location can be related to a lot of other features, everything from local economic factors, local exposures, socioeconomic status. Zip code is used by politicians to target the kinds of ads, and that's an increasingly sophisticated way to get people to vote the way you want. And similarly, it's a way to characterize cancer risk factors. So smoking. So there's a lot of help for smokers using different applications, but there are also new kinds of software which can look at the movement of your arm, and so not only determine the number of cigarettes that you smoke, but how long you inhale, and different features of how you handle the cigarette, which can be related to exposure. Okay, weather and climate. A lot of cancer types are related to features of vegetation, pollution, nutrition, food availability, water availability, insects, dominant air masses. And so incorporating climate will be something on the population level that will be important. And finally, circadian variation. So most of you are aware that there's been a literature for decades that has looked at circadian variation and effectiveness of chemotherapy, the field is called chronotherapy. But also, people that have disrupted circadian rhythms are thought to be at increased risk for a variety of conditions. Again, obesity is prominent, but breast cancer is one that's been frequently mentioned. And in animal studies, when you do various things to disrupt the circadian rhythm, you get implanted tumors that do better. So IARC has determined that disrupted circadian variation is a risk factor for cancer, but these studies in human populations tend to use groups that do shift work or have their circadian variation disrupted by like airline stewardesses or individuals who are blind and lack the diurnal cue from sunlight that resets the circadian pacemaker. So recently we have proposed that a way to determine where you are in terms of body time is to assay metabolomic markers. So we're currently doing a study where we collect metabolomic markers at different times of the day in individuals in a lab, and we hope to use that as a more precise marker of circadian variation. So that is probably enough, and I will stop there, and I'm very happy if anyone has any questions. Thank you. So I didn't say a word about epigenetics, but clearly epigenetics is a bridge between genetics and the environment. And there's a lot of studies ongoing to look at the methylome and a variety of indices of epigenetic changes. So typically in these integrative studies that use tissue, epigenetics is incorporated into it, and it's increasingly going to be important. A recent example of a study like that used methylome data and genetic data to identify MEQTLs that were related to 4 of the 5 top GWAS hits in lung cancer. So they seem to operate by altering methylation. So that's just one example, but there's a lot of studies that will increasingly incorporate epigenetic information. So I'm sorry I didn't mention that. So there's a little bit of a paradox and something we don't understand about lung cancer in other countries. Frankly, it hasn't been studied so much. They often discuss the Indian paradox, where there's a lot of vegetarian diet, and yet rates of heart disease is very high. There are other cancers that are more common in India. It's a similar situation, perhaps, to China, where there's pollution is a major source of lung cancer, but the relative risk associated with tobacco is lower. So current smokers have a relative risk close to 20 in the United States, but in China, it's considerably lower, 3 to 5. And I think in India, I'm not so familiar with all the details of India, but there also has been a dearth of good population studies in India. There really needs to be a lot more research in that area, particularly to dissect the dietary components, because there is such variation. I mean, there's some vegetarian, but there are also a lot of diets that more resemble the West. And so people have said, well, it could be a lot of the spices that are used that convey some protective effect. I think it's just understudied and needs more work. Yes? Okay, so a number of years ago, there was a large study which looked at cell phones and glioblastoma risk and didn't find anything. A lot of these studies are difficult to do. They've also done a power line study, which, because it's an area that's frequently brought up, that low power electromagnetic radiation could have an effect. But most of the time those studies have been negative. They've been criticized, but they've been null. Anyway, thank you.
So we have an announcement, and that being after this next lecture, we're going to be going over to the pathology core in Building 10. So I'll be walking people over if any of you are interested. And we have a schedule change. We're going to have Perwez Hussain today talk about pancreatic cancer. So I'll be talking about lung cancer a little later. And Perwez's title, Pancreatic Cancer, Current Understanding and Future Challenges. Perwez.
Thank you very much, Terry, for switching your lecture with me. I really appreciate that. So I'm Perwez Hussain and not Terry Moody, and I will tell you a little bit about. So what I'm going to do today is to give you a brief overview of pancreatic cancer and discuss with you some of the current challenges and current understanding and future challenges to improve disease outcome in the patients with this lethal malignancy. So let me start with this slide, which shows some of the grim realities that are associated with pancreatic cancer. For example, it's the fourth leading cause of cancer deaths in the United States. The median survival in advanced cases less than 6 months, and there is an estimated 46,420 new cases and 39,590 deaths in 2014 alone. So as you can see that everyone in 2014 who is diagnosed with pancreatic cancer, majority of them will die within the same year. And there is no effective treatment that is available for treating pancreatic cancer. A recent estimate that was reported in Cancer Research has further indicated that pancreatic cancer is going to be second leading cause of cancer-related death by 2030, as you can see here, surpassing liver and colon cancer. There are several risk factors, and that includes exposure and also inherited syndromes and family history. And one of the major risk factors is smoking. And it has been shown that smoking strongly associates with the risk of getting pancreatic cancer. In addition, diabetes, non-hereditary chronic pancreatitis, obesity, and also non-O blood group. This study was done in a division of cancer epidemiology here at NCI, where it was shown that the people with non-O blood group, they are at high risk of pancreatic cancer. And then in relation to genetic syndromes, hereditary pancreatitis, in fact, enhances the risk of getting pancreatic cancer by 50 folds. And also alteration in P16, as you see in familial atypical multiple mole melanoma syndrome, hereditary breast and ovarian cancer syndrome with alteration in BRCA1, 2, and PLB2, two genes, and also Peutz-Jeghers syndrome that has an alteration in serine threonine kinase. In addition, HNPCC, Lynch syndrome, and alterations in the MLH1, MSH2, and so forth. So what we have achieved so far in terms of treating advanced pancreatic cancer. So again, it's a disappointing progress in the treatment of pancreatic cancer. In 1997, more than a decade ago, gemcitabine was approved as a standard care of drug over 5-fluorouracil, which just enhanced the survival in advanced cases just by a month. And then after a decade, erlotinib, which is a EGFR inhibitor, was used in combination of gemcitabine that increased the survival only by 1.5 weeks. But the situation is so bad that it was approved by FDA. And then recently in 2011, a combination of 4 chemotherapeutic drugs, which is called FOLFIRINOX, improved the survival over gemcitabine by about 4 months or so, but it was highly toxic. But recently, last year, a combination of albumin-bound paclitaxel and gemcitabine showed better survival as opposed to gemcitabine alone, but only by 2 months. But this was less toxic than FOLFIRINOX. And they, in fact, used the information from the tumor biology that stroma expresses a high level of a spark protein, and albumin goes and binds to a spark, and so the drug is available to tumor. So as we know that dismal outcome in pancreatic cancer is because of the late diagnosis and poor therapeutic response. We don't have any early detection marker that is specific and sensitive enough for pancreatic cancer. But in about 18 to 20% of the cases, they are identified at early stage and qualify for surgical resection. But even in the resectable cases at early stage, the median survival is less than 2 years, and about 80% of these resected cases, they show recurrence within 2 years. Among resectable cases, about 12% of these resectable cases, they do survive for 5 years or even longer. And there are some clinical prognostic factors that are associated with survival. For example, stage, grade, lymph node, spread, and resection margin. However, these prognostic factors are not consistent, because sometimes you will see that a stage 1 patient resected, resection margin 0, but they die in 2 months. So one of the hypotheses is that there are molecular differences in tumor that determine patient's outcome. So how we can improve the outcome in pancreatic cancer patients? So obviously we need effective therapy that will require the identification of novel effective targets, molecular subtypes, so that a specific therapy can be used for that specific subtypes, and that will include treatment selection or precision medicine, and also improving drug delivery, because I will show you certain examples that have taken advantage of improving the existing drug delivery to the tumors, and that have done better in at least animal models. And then I will discuss a little bit more about that controversy. And then we need early detection marker, and also early detection marker that can identify precancerous lesions and not the cancer, because once the cancer is developed, again, the survival is extremely low. So to achieve this, we need to understand the pancreatic tumor biology, which is the key to improving disease outcome. So let me tell you a little bit about pancreatic tumor genetic alterations and the precursor lesions. So there are several genetic alterations. One of the most important one is KRAS mutation. You see in about more than 90% of the pancreatic tumors have activating KRAS mutations, and about the same 80 to 90% have the P16 alterations. And in about 50% of the tumors you see 50 to 70% P53 mutation and about 50% SMAD4 alterations. So there are several precursor lesions, but the most common precursor lesions are pancreatic intraepithelial neoplastic lesions, or PANINs, and they have been categorized as PANIN1, PANIN2, PANIN3. And there are different timing of alterations that you observe in these pancreatic precursor lesions. So one of the other important characteristic, which is quite interesting in pancreatic cancer, is a huge stroma. So he has a huge inflammatory stroma, as you can see here in H&E sections. So these are the pancreatic tumors, adenocarcinoma, and all these are embedded in a huge stroma. And this stromal cell cells constitute fibroblasts, pancreatic stellate cells, endothelial cells, immune cells, and adipocytes. And it expresses a number of different growth factors, cytokines, and chemokines, and that are responsible for a very active tumor-stromal interaction. The other important feature of pancreatic cancer that it is highly heterogeneous. So this study from Bert Vogelstein group a couple of years ago have shown when they analyzed the genetic alteration in about 27 tumors, they found that there were at least 68 on average genetic alteration, major genetic alterations in each tumor, and that constitute the alteration in about 12 core signaling pathways. And also, each signaling pathway was altered not because of just mutation in the same genes in different tumors, but they had different gene alterations which altered that particular pathway in 2 different tumors. So it was highly heterogeneous. So one of the hypotheses is that, so do we have different subgroups based on the gene alterations or gene expression? So this analysis that was done by Causation and colleagues, they analyzed the gene expression in about 27 or so tumors, and they found that based on the gene expression, they subdivided the tumors into 3 subgroups, and what they found that they in fact had different survivals. And also when they did some of the in vitro studies where they have the same gene expression as these 3 tumor subgroups, they responded differently to the standard care of drug gemcitabine. One other interesting observation in pancreatic cancer, as in other tumors, they alter the tumor metabolism to fulfill the requirement of the energy need for the tumors. And in pancreatic cancer, there are several alterations or reprogramming that have been reported. One is that increased glucose uptake and also enhanced glycolysis. And this glycolysis is used to activate some of the enzyme of the non-oxidative pathway of phosphate shunt that enhances biosynthesis. They also have enhanced glutamine absorption, and they use glutamine not only to fuel the TCA cycle, but there is an also other alternate pathway for making a higher amount of pyruvate and NADPH, which they use for maintaining the redox status for maintaining the tumors. So I won't go more into details on the metabolic parts. And what I would like to go a little bit more in details about the complex stromal network, because a number of attempts have been made to target stroma to achieve effective treatment. So let me just give you a brief overview of what happens in the pancreatic cancer stroma. These are the tumor epithelial cells, and then there is the mesenchymal cells, extracellular matrix, and hematopoietic cells. So what you see that there is a huge immune suppression, and immune suppression happens because of the overwhelming presence of the T regulatory cells, tumor-associated macrophages, and also myeloid-derived suppressor cells at the tumor sites. And these immune suppressive cells, they inhibit the CD8 T cells, and they inhibit the anti-tumor immunity. These tumor epithelial cells, they secretes a number of different cytokine and chemokines that supports the migration and attraction of these immune suppressive cells. Then there is a pancreatic stellate cells, which is activated by the platelet-derived growth factors, FGF, sonic hedgehog pathway, and TGF-beta that are secreted by tumor epithelial cells, and these are very important in maintaining the huge desmoplastic stroma. And one very interesting thing is that in these extracellular matrix, there are so much accumulation of hyaluronic acid that it enhances the interstitial pressure on the blood vessels, and blood vessels are compressed, and there cannot be transfer of the chemotherapeutic drugs. And I will show you one example that what happens that when they relieve the interstitial pressure, the drug went into the tumor, and then they achieved better survival. So treatment strategies to improve disease outcome. So first is the drug delivery and effectiveness of systemic therapy and the targeting of a stroma. And most of these studies have been done in genetically engineered mouse model, which involves the mutated KRAS and mutated P53 that are specifically inactivated and activated in the pancreas. So there is a very common genetically engineered mouse model, which is known as KPC model, that was developed by Sunil Hingorani, as you can see here. And this faithfully recapitulates the developmental stages and progression of pancreatic ductal adenocarcinoma in humans. So what they did that in one of the studies by Ken Olive, they inhibited hedgehog signaling, which depleted a stroma, enhanced drug delivery, and improved survival in mice. So as you can see here, that the mice that got hedgehog inhibitor, IPI 926, and also gemcitabine, they had much better survival as compared to those, this red curve, that got only gemcitabine. So they were able to enhance drug delivery by depleting a stroma, and the end result was improvement in survival. But unfortunately, this clinical trial has failed, and it has not improved survival in patients. So now coming back to decreasing the interstitial pressure on, as you can see here, because of the accumulation of hyaluronic acid, there is an interstitial pressure which is much higher than the vascular pressure, and it is compressed. When you use hyaluronidase, a PEG-PH20, it relieves that pressure, and you can see the extension of these vessels, and the drugs can go outside into the tumors. And here they have used both gemcitabine in addition to the PEG-PH20, which is hyaluronidase. And what they found, again, is an enhanced survival in the mouse that received both gemcitabine and PEG-PH20. And this trial is ongoing, and phase 1B trial is successful, and this PEG-PH20 is categorized as the orphan drug by FDA. So hopefully we will see some better outcome with this approach. But it's not that simple, because stromal targeting may not always have beneficial therapeutic response. There are two papers in Cancer Cell in 2014 that showed quite contradictory results. So one paper came from Andrew Rim's lab here in Cancer Cell, and what they did, they also used genetically engineered mouse model. They inhibited sonic hedgehog pathway, but what happened is that it became more aggressive, and they concluded that sonic hedgehog as a tumor suppressor in pancreatic ductal adenocarcinoma using these genetically engineered mouse model. And you can see here survival from palpation of tumors. again low. Here, macrometastasis is high, and also acinar to ductal metaplasia is high, and also, so the number of pannin lesions, they were higher as compared to the PKCY mice. Yellow, they have used the fluorescent marker to trace different cells. Then another paper that is consistent with these findings came from Raghu Kalori's lab by Ozdemir and colleague, and they used a genetically engineered mouse model where they depleted the myofibroblast that was expressing smooth muscle actin. So expression of a smooth muscle actin is the indicator of activated myofibroblast. And they also found that both in early pancreatic ductal adenocarcinoma and late pancreatic ductal adenocarcinoma, they found high pathological scores here. Here also, its tumor necrosis was high, as you can see here. In the depleted and in the late, there are more poorly differentiated tumors. So they also found that the mouse had worse survival, where the depleted myofibroblast, stromal myofibroblast here. So tumor-stromal interaction is complex, and therapeutic approaches targeting stromal needs caution and may require new molecular taxonomy in pancreatic cancer. So it's complex and needs more investigation. Maybe there will be some tumor subtype that will benefit from a stromal depletion, but some may not. So let me give you the last part of my talk. So I will not go all the way for 1 hour. And that is the association with inflammation. And this association comes from several observations, both epidemiological and molecular studies. One is that, as I mentioned, chronic pancreatitis enhances the risk of pancreatic cancer. You see a huge inflammatory desmoplastic active stroma, which with a lot of active inflammatory cells. It always has a constitutively active NF-kappa B signaling. You see a number of different inflammatory cytokines that occurs with a higher level, and it also shows an increased expression of COX-2 and NOS2. We also have evidence of a stepwise accumulation of inflammation during development and progression of pancreatic cancer. These are pannins, and in pancreatic ductal adenocarcinoma, you see the accumulation of inflammatory changes. A very interesting study by Craig Loxton from MD Anderson in Journal of Clinical Investigation have shown that it's not only the mutated KRAS, it's the pathologically active level of mutated KRAS. And what he suggested that all the mutated KRAS does not have the same oncogenic potential unless it is maintained at a level by inflammatory mediators. And what he suggested is that, and he showed evidence that there is a feedback inhibitory loop through the activation of NF-kappa B and the generation of inflammatory mediators like prostaglandin that maintains the high level of KRAS, oncogenic KRAS activity. So I will tell you a little bit about one small piece of work that we are doing in our laboratory at the National Cancer Institute. So MIF is an inflammatory mediator. It's a pro-inflammatory cytokine. It is expressed in epithelial cells and inflammatory cells, and it regulates immune and inflammatory responses. Increased expression is found in many tumors, including pancreatic cancer. It enhances a number of different oncogenic signaling pathways, for example, NF-kappa B, AKT, ERK1, and ERK2. enhances COX-2 and NOS2 expression, and also it inhibits P53 and RBE2F pathway. And based on these and several other functions of MIF, it is considered as a connecting link between inflammation and cancer by several different labs and investigators. So the hypothesis which we tested is that MIF contributes to pancreatic cancer progression and predicts disease outcome. And what we found is that, first of all, we saw that tumor expressed a higher level of MIF as compared to the normal appearing ducts in adjacent non-tumors. And what we also found that the tumors that expressed a higher level of MIF, the patient had a shorter survival as compared to those patients where tumor had a low level of MIF. And this we also analyzed by both univariate and multivariate analysis and showed that MIF predicted the survival independent of other covariates. And then we validated these findings in other cohorts, and again you can see the higher MIF expression is associated with poorer survival in these pancreatic ductal adenocarcinoma cases. So this was an association. So what MIF is doing? So one of the important events in pancreatic cancer progression and cancer in general is the epithelial to mesenchymal transition. And again, Andrew Rim in his Cell paper in 2012 showed that inflammatory mediators in pancreatic cancer, and he used the same genetic engineered mouse model, can enhance epithelial to mesenchymal transition. So we tested the hypothesis that MIF induces epithelial to mesenchymal transition in pancreatic cancer. And we did some in vitro studies using the pancreatic cancer cell by expressing MIF in these cells. And what we saw, this is the QRT-PCR, that we saw a decrease in ecadherin and increase in vimentin, which are the marker of EMT. And this is the Western blot analysis. And then further visualization of ecadherin and vimentin, as you can see in the MIF overexpressing and controlled cells. So then we went a step further. We used the orthotopic mouse model to see what MIF overexpression does, and we saw an increase in metastasis and tumor growth in these orthotopic mouse model with MIF overexpression. And interestingly, when we looked at these two tumors, so the MIF expressing tumors was poorly differentiated, every single tumors, and the control were well to moderately differentiated. So we did gene expression analysis, and we found that MIF induced a change in global gene expression profile, and that further showed that MIF overexpressing tumors showed expression of EMT-related genes, as I showed you in the cell lines. So what happens when you remove MIF from a genetically engineered mouse model of pancreatic cancer? So that was our next question, because the idea is, can you inhibit MIF to treat pancreatic cancer? So first step was this preclinical model, and as you can see that it expresses high level of MIF in the tumors. We generated MIF-deleted KPC mice that, of course, doesn't express MIF. And what we found is that MIF deletion enhanced the survival, significant enhancement in the survival. So now we are investigating the mechanism, that what is happening, what MIF was doing, and what MIF deletion does that enhance the survival. And the next step is to first let the mouse develop the tumors and then treat the MIF with, and then inhibit the MIF by using different inhibitors. So there are different inhibitors that are available, including the small molecule inhibitors and then anti-MIF antibodies. And currently, anti-MIF antibodies is in clinical trial for several different solid tumors, phase 1 trial. And Dr. Bukala have a small molecule MIF antagonist, and I'm collaborating with him in the preclinical model to see if the small molecule MIF antagonist can have any effect on the pancreatic cancer progression. So in summary, what I have shown you in our studies, that a higher MIF expression is associated with poor outcome. MIF induces EMT in pancreatic cancer cell line. MIF enhances growth and metastasis of tumor xenografts. MIF deficiency increases survival in KPC mice with lethal pancreatic ductal adenocarcinoma. MIF may be a candidate target for designing improved treatment. And I would like to again end with this slide, which I started with, that we need to understand tumor biology to identify better detection marker and the effective therapy by identifying novel targets. So I would stop here, and I would like to thank you for your attention, even as a second talk after 1 hour talk, and I appreciate that, and I will be happy to discuss you with any questions that you have. Thank you very much. Yes, please. Yes, that's it. That's an excellent question. In fact, we are doing that. So what we did, we went back to humans then, and then we subdivided the tumors in 2 groups, one with high MIF and one with low MIF. And then we have done the analysis for the gene expression, mRNA analysis, Affymetrix gene array, microRNA analysis, and also metabolomics. And we are currently analyzing those data, and we are finding some candidate interesting genes and pathways. Yes. Yes, so that's again a very good question. So it is found in the tumor cells, epithelial cells, and also in many different cells, inflammatory cells and immune cells. So one of the important questions that need to be asked, that whatever I show you, it's all autocrine effect, that the tumor expressing MIF and you express MIF in tumors or delete MIF in tumors. But we have to see what is the paracrine effect coming from the inflammatory and immune cells. We will be doing those experiments. Yes, please. No, I haven't, but that is one of the hypotheses which we are interested in. Yes, all these are very good questions. Thank you so much. And I'm happy that we are going in the right direction with the discussions and the questions that you have asked. Thank you.